name is my name is Esha, and I'm just across the street. I'm studying biology and film. It's my undergraduate at Webster University. And my research is part of the required senior thesis we're supposed to do our senior year. So I am a junior, so I do have time. Technically, credit-wise, I'm a senior, so I could graduate, but it's my third year at Webster. So this can go for 2 years, it can go for 1 year, depending on the research I collect. And so it will be covering the effect of zooplankton on the buddens of BD, which is a citric fungus affecting all types of amphibians. But the amphibian I focus on is frogs. Okay. Okay, so in a lot of my Webster classes, my freshman and sophomore year, we talked a lot about the sixth mass extinction that was going on, and that really piqued my curiosity. I wanted to know what exactly was going extinct and why was it happening. And what I found out was the only major group that was globally going extinct was amphibians. And so I found this biomap on an organization called AmphibiaWeb. So in the red, you can see the number of extinct amphibians extinct in the wild are critically endangered species. And the orange is the number of endangered or vulnerable species, and then white is the total of species within the biome. So globally, we are experiencing an amphibian decrease, extinctions. And what is this major driving cause, I was wondering? And the major driving factor is BD, a deadly aquatic fungus. So how does BD work? BD lives in the water as a low zoospore, and it travels through the water and reproduces until it can find a host. And these hosts are commonly amphibians. So these BD burrow into the skin of amphibians and create lesions in their skin so they can't breathe anymore, which kills them. And inside the host, they start reproducing and more spores form, and then the spores get into the water, and the cycle continues and keeps killing off these frogs, salamanders, other amphibians. And the cycle will just continue until the habitat gets wiped out, they go extinct, and then the BD will just stay in the water and travel downstream and find another group of hosts to infect. So previous studies that I researched, I looked into a lot of literature, but these were the two that affect my project the most. So one was 2013. It was a Missouri study down, I think, and the Tyson Research Center, so through WashU as well. And they tested 29 ponds, and 11 of the 29 ponds were positive for BD. So the question was, why doesn't BD occur everywhere? Why isn't it in 29 ponds? And so they tested a lot of different factors. Factors included the pH of water, the macroinvertebrate diversity, and how much the density, how many tadpoles were in the water, how many frogs were in the water. And unfortunately, so this was only a summer-wide program, and they couldn't find the ponds where BD was found were not statistically random. However, no biotic or abiotic variables statistically were significant. So there was no conclusion that came out of the paper, but they did know that these occurrences weren't random. So at the end of the paper, they kind of suggested some things of why this was happening. And one of the things that they suggested was predation on BD. And I found another paper, which was a 2011 paper, which was that water fleas can actually eat zoospores. So water fleas were starved for 24 hours and then exposed to BD, and then after 3.5 hours, the guts of the fleas were exposed and BD was found within the fleas. So my thought process was, if there's zoospores, zooplankton in the water, and the zooplankton are eating these zoospores, maybe that could be one of the reasons that BD isn't found in all these ponds. Which led me to my procedure. So I have not researched yet. I plan to do it this semester. Going to next semester, BD is found most prevalent in ponds in the spring, winter seasons. So first, I will find 10 ponds and identify 10 ponds, locations, and the permissions needed to go to these ponds. And I'm doing this through the help of the zoo and the faculty at Webster. And then I'm going to be collecting zooplankton. So there's 10 sweeps of a zooplankton net in the ponds at 5 different locations at each pond, so 50 sweeps in all. And once we've collected them in the net, we'll take them back to Webster and look under a microscope and identify them. Then we're going to preserve the zooplankton until they can be taken back to Webster in Lugol's iodine solution. And we'll be swabbing tadpoles. So 30 tadpoles will be swabbed with a focus on the mouth because the mouth is where BD fungus is found, and these lesions occur in the tadpoles' mouths. Then each swab will be properly sealed and ran through a BD DNA detection test to see if these tadpoles were BD positive. And we will identify zooplankton, as I discussed, with a compound microscope back at Webster, and the results of the BD detection and zooplankton identification will be recorded and analyzed. And my hypothesis, as I discussed, the lower the zooplankton density, so lower zooplankton that are eating these BDs, the less tadpoles that will be infected with BD. So my question then is, how does the zooplankton community of a pond influence the number of BD-infected tadpoles in the environment? And so again, my hypothesis is that the way they'll be affected is the more zooplankton, the less BD. And so how is this affecting nature, and what is the benefit of my research? Citric disease is believed to be responsible for one of the most significant disease-associated losses of biodiversity in recorded history. There's currently no viable treatment or vaccine for amphibian infected by BD. So this is something I found really interesting when I was actually reading papers. I kept trying to see if there was treatments or there was some way to cure frogs that had been infected once they get infected. But so far there is absolutely no treatments that have worked, and there's no way to help these frogs once they've been infected. So it's crucial that we continue to research on this disease to prevent its spread and the decline of the amphibian species. And my research attempts to draw correlations between previous studied factors to gain a clearer understanding. And so this is how my budget will be used. We already used some of the budget to buy these swabs. We'll also be buying a plankton collection net to collect the zooplankton, different permits, and then we'll be sending off the swabs to an outside lab to get it tested for BD, and then mileage, we're just driving between 10 pounds is a lot, and gas is expensive, so that's what I'll be using that for. And my support system at Webster, the lovely Dr. Stroutman is actually here. So she's my faculty advisor. She's been helping me for a year and a half now, so this has been going on for a while. Dr. Kim Kleinman, he is my academic advisor. Dr. VBK is actually working on a barcoding project, and so we are in discussion if we can also eDNA the ponds that I research at to see if BD zoospores can be barcoded in these ponds. And then Mike Dawson, who some of you might know, but he is the coordinator of the Wildlife Spring Peeper program. So he was helping me identify ponds and swab these tadpoles. And then I have two funding places, obviously Vignes, which is the Mikey Scudder Scholarship. And then I also have a collaborator grant from Webster University. And then my friends and family, that's my family up there. And so here are the references. If you guys would like me to send this presentation so you can read more on it, let me know. My email is up here. Thank you guys. Any questions? My first time presenting all this in front of a group, so I apologize, I'm a little nervous, but yes, go ahead.
I always like it when somebody comes up with a different idea that or read about it. Actually cause the fungus to, get rid of it, control it, because it's kind of unique. I have not heard of it, but I applaud you for that. Obviously not expert on the field, but it's not generally known. I know. Look at that. I'm going at
yeah. Yeah.
So,
yeah.
So I'm going to wish you luck. This actually shows some chances of actually working. this could actually bring up needs to help the animals. They are serious.
Yeah, thank you so much. No, when I stumbled across the paper that these water fleas were eating and digesting the zoospores, it blew my mind. So I was like, why we should look more into it. So yeah, it was interesting to find. Anything else?
Good luck with that. That's what keep working at it.
Yeah. Any other questions? Yes.
Yeah, so you don't have your 10 ponds yet, or is that still in progress?
It's still in progress. I have some ideas, but we are still looking for them. That was one of the ponds we were looking at, yes. Tyson, and then they have a pond down at the zoo that Mike Dawson was also looking at. So those are two main options right now. Oh yeah. Oh yeah. Yes.
Not heard from fungus.
That's it's everywhere. It's a global issue. It's primarily affecting the amphibians in Australia, so that's where the fungus is found. But it's all across the world. There was papers from India, there were papers from Korea, there were papers from North America, South America. So it's a global fungus. Yeah. Yes, so it's not native from here, and there are debates as to where it originated from. A lot of papers say Asia, other papers say Africa. So it's not sure where it originated from, but it's not North America, from as far as I can tell. Mm-hmm. Yeah. Mm-hmm. Yeah. So there's two strains. There's a strain B Sal that affects salamanders, and then there's another strain that primarily affects frogs. And so like these kind of merged and then like came to North America and is now affecting both salamanders and frogs at the same time. So
wherever the fungus came from, wherever amphibians are living near the should get the should come on there.
Wherever we were in the last few strains, they share like the shared genes are pretty readily.
Okay.
So if you have, it'd be like COVID, right? Strains of COVID, and all they're just they didn't want they come together, and all the genes together. Right? Something similar might have happened, where you get two different strains that were able to create a new strain. No species on it. There's lots of other not saying it's just some evidence for that out there.
yeah.
Conjecture.
The new strain is in the past 30 years, but BD in general has like been around for according to all the genotypes and the genetic papers I read for a while.
But there are no areas of the world that amphibians are
Completely adapted to? No. No. Not from what I found, no. No.
So remember that they found that was like a higher altitude or something.
Yeah, so they researched it was a place in Argentina where they found ponds that were like in a valley and then found ponds that were higher altitude, and it did have a slight effect. I don't remember which one, like what exactly was affected, but I do remember there was a significant difference between the two. Yeah. Yeah. So Smith actually, the 2013 paper, one of the things he did test was the temperature of water, and temperature of water, along with the other variables he did test, were not significantly correlated with anything. But it was just like, I think it was undergraduate research that only went like for a year, so there wasn't a lot of data. So I don't know, maybe temperature does affect it, but the 2013 paper didn't find any evidence. Yeah. Anything else? Anything for chat from Zoom?
Mr. Comment, that all three presentations were excellent.
Okay, thank you guys so much.